So we will continue with this history of what's happening. They are in the blacklist for all over the life period. I don't know the real. Yeah. You know macumba. Macumba is something that we can do. It's very bad for people. We made a small puppet and we put voodoo, and we put some pins. I don't recommend you. That's true. Well, so I will finish what I started yesterday concerning this different type of model, which we call here the thermal lens model. So what we are doing now, we are observing the modification of the index of refraction of a sample due to the modification of the temperature of the sample. So essentially this is what we expect if we take into account what is happening in a sample which is shining with a laser beam, for example, in the time scale, not now from the femtosecond regime, but now with a larger time scale, typically milliseconds, tenths of milliseconds. So when you shine your sample with a laser during this time, what happens is a modification of the density of the sample locally. And this modification of the density depends on the intensity of the beam you are working with. So basically you may think that you have essentially a lens, which is a thermal lens. This is the focal distance of this length. The optical path length, if you have a sample of thickness B, the optical path is essentially the thickness of the sample you are investigating, multiplied by the index of refraction. But now the index of refraction has the linear part and the nonlinear part, which is proportional to this term, which is the term here, is essentially the thermal optical coefficient. So we are investigating now a different type of physics. We investigated just now looking the electronic modifications of the lens. Well, if we are dealing with the heating of the sample, this is basically the diffusion equation for the heat, the temperature, the density of the sample, specific heat, the heat there, the divergence of this term. So essentially what we have to calculate is the amount of heat that you will pump in your sample during the time where the pulse is interacting with your medium, which can be a complex fluid medium, solid medium. It must be only transparent to light. And with this, we are able to calculate what is important here is essentially the variation of the temperature, which depends basically on the absorption of the sample here, the power of the beam. This is the typical time for the transmission of heat. As I told you, is about tenth of milliseconds. This is the time. R is essentially the coordinate, is the center of the beam, the radial coordinate here. And omega is something which depends, was in the previous transparency, is essentially the radius of the beam in a particular Z position. Okay, remember that we have a cylindrical beam. After that we put a lens, a glass lens, which will focus the beam and open. So the waist of the beam depends on the Z position. Okay, so this is written here. So the dependence on Z is present in this particular radius, which is located in this position of this formula. So this is the modification of the temperature of the sample when you shine it with a particular laser beam. Only to have an idea of orders of magnitude, we can have a difference between the center of the beam and the border of the beam, which has about 20 microns of diameter. You have a modification of temperature of about 5 to 6 degrees. So it's possible to have this type of modification of the temperature from the center to the borders of the beam. So it's possible to see the modifications in the density of the sample. And also, if you leave this for more time than the typical thermal time, you are able to measure the Soré coefficient. Okay, so we establish a temperature gradient there from the center to the border of the beam, and the Soré effect will take place, and after this we can see a matter lens which is formed in a different time scale. Well, after that we can calculate the focal distance as a usual lens, taking into account the sample thickness, the modification of the temperature, and we have this parameter here, which is essentially a dimensionless parameter. We'll see in the next transparency now. This is the intensity as a function of z, position, and the time. And this parameter here will bring all the physics we can extract from this experiment, which is basically the thermo-optical coefficient. All the other parameters are calculated or measured independently. So for example, here is the linear absorption. You can measure independently. This is the sample thickness, the power of the beam. Pi is pi. Omega naught is essentially the waist of the beam, the smallest waist that you have in your beam. So the waist in the focus, at the focus position. So if we are able to measure this parameter here, which enter in the expression of the intensity, the normalized transmittance, if you divide this by that, you have theta as a single one fitting parameter. So it's a single one fitting parameter that you have, and you are able to calculate this dn over dt. So the normalized transmittance can be written in this form here. As you see, you have only this fitting parameter. If you measure this theta, you have the thermo-optical coefficient. So this is something. Pardon? Someone asked it? No.
How do you determine the absorption coefficient?
In a spectrophotometer, it's a linear absorption. As I showed yesterday, remember, we had a figure like that, a graphic like that. In the case of ferrofluids, remember that you have alpha as a function of the wavelength, was something like that. That's the alpha that we measure in a spectrophotometer. Okay, so you choose the wavelength you want, so this alpha comes directly. Well, in the Z-scan technique, we'll show we'll make the experiment together now. Well, this is the oscilloscope. We have a laser beam here. This is a chopper, okay? What a chopper is essentially a disc with some holes in the disc. So when we turn the disc, sometimes we have the beam, sometimes you don't have the beam. With this, we are able to have beams pulses about some tenth of milliseconds, okay? So this is the chopper. This is a beam splitter that will take part of the beam to this detector, and this detector will be shown for you here in blue. Is what it detects during a pulse. This, you'll forget, it's not important. Now you have the lens, which will take the beam that pass through the beam splitter, will make the focus at this position approximately. After that, we have another detector here, which is connected to the other channel of the oscilloscope. And here you see the iris. So with this setup, we can measure N2 or dN over dt. Okay? So this is what we measure here in the oscilloscope. So during this period of time, the chopper is closed, chopper is open, you measure this, after closed, okay. Is it okay? Well, this is the Gaussian beam. If you make a cut perpendicular to this direction, you have more intense beam in the center and less intense beam in the middle. Up till now, it was essentially cylindrical. Passing through this glass length, it will converge at this position, and after that it opens. And what we measure in this detector here, which is the blue detector, is essentially what we see in the red detector. Okay? Yes? You can use a lock-in, yes, yes. The lock-in is very useful when you want to remove the background light. So this is very useful. In the lab we have this, but I think here to show you, somebody could not know exactly what is a lock-in, so the oscilloscope probably is more good. Well, so let's see what happens with a sample with N2 smaller than 1, or dN over dt negative. It's the same thing in the thermal regime. If we have the sample now far from the focus point, there is no effect, no thermal effect present, so you see exactly here as you have a linear sample. Nothing happens. If you go near the focus, you see now that the transmittance now increases as a function of time, and after that, if I have a larger pulse, it will be absolutely constant. In other words, this concentrates the light in a smaller place in the iris position, so we have more photons here, more intensity, more transmittance is measured, so the thermal lens is being constructed. And after this time, it reaches the equilibrium state.
Excuse me, is this data this is actual photographs of the oscilloscope, or this is...
No. I will show you what we have after in an actual experiment, the next to transparency, I think. And when you go after the focus, you have some type of divergent lens, so the beam is opened, less energy comes through the iris, and you have a decrease of intensity as a function of time, so the lens is deformed. And when it goes very far from the focus point, we recover a flat signal here. Okay, so this is essentially a Z-scan experiment. This is what we measure in the different time scales. If you are interested in electronic process, you make this pulse in femtosecond time scale, thermal millisecond time scale, or SORE, for example, in second time scale. For you. This is a real experiment, okay? So we see here the normalized transmittance as a function of time. So this is the formation of the thermal lens. The time scale is in milliseconds, so 20 milliseconds if I see well. Here is exactly at the focus position, and when you cross to the other side, you have the deformation of the thermal lens. So this is essentially what need to be measured to construct this Z-scan typical curve, okay? So with this, we are able to calculate what we want. In other words, you can say that when you have the chopper in the on position here, you hit the sample. After that, in the off position, the temperature of the sample decreases, and so on. So we can measure a lot of different pulses to have a good statistics, and after that, we are able to get good numbers after that. Well, for the SORE, I promised you yesterday, this is the picture of the guy with a very nice mustache. I think I recognized yesterday him in one of the walls of the scenario. His scenario, yes. It was a picture, looks like him, but probably he was here in one of the first schools you organized it. So the idea is that, as I told you yesterday, if you have a temperature gradient, it will impose a gradient in the concentration of the different components of a mixture. For example, if you have water and alcohol, you put a temperature gradient, the molecules of one of this will concentrate in one side, the molecules of the other species will to the other side, so it imposes a concentration gradient. And we have the exact reverse effect, which is named Dufour effect. If you establish a concentration gradient in a sample, it will impose a temperature gradient in the sample. It's a very interesting phenomenon which was discovered by Charles Sauvé, about 1860, about, so more than 100 years. Well, this is what I told you before. It was discovered around this period here. And we have these two possibilities in the case of materials, general materials. Ferrofluids, for example, we have both types of materials depending on which type of ferrofluid we are considering. We have the thermophobic here and the thermophilic. In the case of thermophobic, remember that we have a Gaussian beam, so it's more intense in the center, less intense in the borders. The thermophobic, all the particles tends to go to this direction, so we have a radial gradient pointing to the borders. And in the thermophobic, it's here, and thermophilic is here. So it likes the heat part of the sample, so you have the concentration of the particles at that place. Well, as I told you, if you have the possibility to separate in different time scales different types of phenomena, you can construct a general model or generalized thermal lens model where you take into account the different sources of modification of the index of refraction. You have the electronic process modification of the index of refraction, the change in the volume fraction, which is the Soré effect, and the thermal lens effect here. So the optical path length is essentially a sum of three different lengths which will be formed in your sample, take into account that they have different characteristic time scales. This is the sample thickness, this is the linear index of refraction, so everything which is inside this bracket is the index of refraction, take into account the electronic process, the Soré process, and the thermal lens process, which is here. And look that in the case of the Soré coefficient, if you are I will not go into the very details here, but we have a reference that is that reference here. It's this reference in the Journal of Optical Society of America B 2003, where we developed this generalized thermal lens model. And it's very easy for us to investigate these three types of phenomena because they have different time scales. So if you start measuring, for example, the very short physics which occurs in the length of population, you can determine this term. This term is not yet formed because you need time for that. The thermal length is not yet formed neither because you are in femtoseconds time scale, so you can measure this independently of this one. After that, we increase the pulse width and we measure the thermal contribution, okay? The other one we had already. And after that, we increase more the pulse width. And with this, we can successively measure the different contributions to the modification of the index of refraction, okay? So this is the beauty of the model here. So this is what I told you. Thermal lens here, the population lens here. And in this model, we make a generalization taking into account that you have not only a solvent and a solute. You can have more than one solute, as we will see in the case of lyotropic liquid crystals. So if you have, for example, water, molecules, free molecules, and micelles, which can be together in these lyotropics. So we can calculate the Soré coefficient, the contribution of this Soré coefficient, take into account the different contributions of each of the solutes we will discuss. So in the case of the population lens, we are able to define focus distance of this length, which I call here n. N is for nonlinear, but imagine that this n is essentially for the electronic process, okay? It's something that is absolutely, has the dimensions of 1 over meter here, and this is dimensionless, so it's some kind of phase introduced by the population lens. Power of the beam, sample thickness, N2 is what we have to determine, make an experiment, you remember that omega naught here is the waist of the beam. And the characteristic time scale is femtosecond. If you go to the thermal regime, now you have the thermal length contribution, which has also this phase here. We can call this a phase characteristic of the thermal length, which is also dependent only on the thermal optical coefficient. All the other parameters are known. Okay? And finally, in the case of the Sorel contribution, you can have here the same phase. This is the matter length, which depends on a parameter which is more complicated than the other one. And the complication comes from the fact that you can have more than one solute in one solvent. So this sum here takes into account essentially the contribution of the n solvents that you have in your n solutes that you have in your solvent. So this is what we can measure in this experiment. Is this complicated letter here? I don't know the name exactly. But all the other things are measured independently, and the time scale is much larger than the other one. Well, okay, I think I will go to the next. This is not so important. I will, well, so we will see some applications, essentially two applications of these models we discussed up to now. The first one concerns liquid crystals. So we heard about liquid crystals today. So my first question is not to respond at home, but here. What is a liquid crystal? If nobody says, I will choose.
Long particles that can orient or not. That's the simplest description. There are long particles that can be oriented or not.
So liquid crystals are long particles.
The simplest answer would be that can move and can orient in position.
What do you believe? What do you think about that? He's saying that liquid crystals are particles, long particles.
Some sort of structure, maybe?
Liquid crystal is a structure. So particle structure, what's a liquid crystal? No. No. Argentina. Caipirinha coada, always. What's the liquid crystal? It's hard to say. No? I'm doing that not to pressure you. It's not a pleasure for me to do that. I hope that you will keep this in mind when you leave this room. Blue guy. We have molecules here, particles, we have structures here. Argentina is in doubt.
I don't know, maybe I've just thought some rubbish.
Please.
A liquid crystal is something that has some kind of ordering, but not like long range order like a crystal. But it still has some like, if you check, for example, the distribution function, you can see that it has some like peaks, but they just decay with some like maybe exponentials.
Well, but if we start with your phrase, liquid crystal are something.
Yeah, it's something, of course.
It's something.
Material maybe, which
A
has this kind of properties.
Well, a material, particle, structure, Argentina, nothing. What is a liquid crystal? Okay, the liquid crystal is complicated. What is a crystal? Remove the liquid. Pardon? It's something.
No, there's a
Something
periodic array of atoms.
periodic. But we have a name for this, no? speak about crystalline solids, for example. Sodium chloride, something like that.
He mentioned long-range
Long-range ordering. So crystals are long-range ordering.
No,
No.
particles with
Are
fixed
particles.
position.
We fix it in particles. Yeah. Yeah.
It's a repeated structure, which I call particles, and it has all the symmetries and
Yeah.
other dimensions.
Yeah.
Yes, and this.
May I propose a name for this? State of the matter.
Ah,
No?
Solid?
No, crystalline solid. It's a state of the matter. And the isotropic liquid is also a state of matter. A liquid crystal is a state of matter. It's not a particle, it's not something, it's not a structure. It's a state of matter, has the same status of one state of the matter. A mesomorphic state of the matter between the isotropic liquid and the crystalline solid. So it's a state of matter, has this status in the mankind. It's a state of matter between the liquid isotropic and the crystalline state. It presents some degrees of order. Materials in this state of matter present some ordering, which may be positional ordering, short-range positional, we'll see this, orientational ordering. So some degrees of order are present in this state of matter. If you read, for example, the book of Pierre G. Degennes, Nobel Prize 91 in condensed matter physics, you will be convinced that we have really a state of matter. And what is important to present some particular degrees of ordering is that the basic unities of this material should present some type of shape anisotropy. What I mean by shape anisotropy is not only some geometrical aspect, but maybe also the presence of a dipole, electric dipole, magnetic dipole. So these are the basic unities which may construct a structure like that we'll see in the case of liquid crystals. We have two types, two big families of liquid crystals. One of them is called the thermotropic liquid crystals, which are made essentially by molecules, okay? Free molecules, which present the shape anisotropy as we will see. And another one is the family of the lyotropic liquid crystals. This, I think 99.99% of all the technological applications of liquid crystals are due to this particular thermotropics, where the phase transitions occur as a function of the temperature, keeping the pressure constant. And in the case of lyotropics, we don't have technological applications, but the interesting aspect of it is that it has a very big border communicating to biophysics, and we will understand this in a while. Well, thermotropics, for example, this type of molecules which present a very high shape anisotropy here, so this dimension is much larger than that dimension here. We're about 20 angstroms and 6 angstroms in this direction here. It's like a stick. This basic unit is elongated molecules. The name is not so important, but it presents liquid crystalline states between the crystalline solid and the isotropic liquid. Another type of molecule that presents the shape anisotropy is this unpronounceable name here for the physicists, but we call discotic molecules. So very large diameter with respect to the thickness of this cylinder. And between the crystalline solid and the liquid isotropic, you have three different liquid crystal states as a function of the temperature. We can have also some type of molecules which were named banana molecules, which has this type of configuration, boomerang. There are different names, but usually it's considered as banana. So we can change the orientation of these two arms here, changing this angle, and we can have symmetric or asymmetric arms, and as a function of temperature, you have a lot of different phases, okay? Different structures. Lyotropics, on the other hand, are basically a mixture of this type of molecule, which we name amphiphilic molecule, where you have a polar head here and a non-polar tail, which is essentially carbon and hydrogen, which is this part of the molecule is not soluble in water, but this part is highly soluble in water. This is an alcohol which has basically the same property of this soap here, but it is used as a co-surfactant. It's another molecule which we will introduce to impose some nanosegregation, we will see in a while. And it is interesting to produce some kind of nematic liquid crystals, which is different from the nematics you saw this morning. Because this morning we saw uniaxial nematic liquid crystal, where you have only one axis of symmetry infinite. Okay, you can change the orientation by any angle, you have essentially the same energy. In the case of biaxial liquid crystals, you will see that it's completely different. It's the unique example in the nature that you have a liquid with 3 different index of refraction. So you orient your sample, a liquid sample, and the velocity of propagation of light is different in the 3 orthogonal directions. It's common in solid crystals, but in liquid crystals, the only example I will show you today here. So when you mix this type of molecules with water, here you have a high electric moment. Will be a very strong interaction between these two parts and a very weak interaction between the paraffin here, or the carbonic chain, and the water molecules. And what happens if you put enough molecules like this in water, what we call the critical micellar concentrations. So before this critical micellar concentrations, you have this type of molecules in water. They are not happy because this is good in water, this is not good. But if you put more of these molecules, what we will form is some type of basic unities. Imagine a ball of American football, okay? Some kind of ellipsoid, where you have in the surface of the ellipsoid the polar heads in contact with water. And in the interior of this ball here, you have the carbonic chains. So this structure is essentially made to avoid or to minimize the contact of the polar solvent with the carbonic chains. Okay? Well, we make things very comparable to this every day. You can give me an example of what we construct, what we build every day with this type of molecules in our day life. Probably we did that.
Soap.
What?
The soap.
Soap, yes. Why you have, for example, oil in the hand? If you put only water, it will only smash the oil in all your hand, nothing will happen. But if you put soap, that's okay. You will form a big structure and the oil will be inside and you can wash your hands. Okay? So it's something very similar to that. And not essentially micelles that we will form, but the principle is almost the same. This is an example of a lyotropic liquid crystal which presents as a function not only of the temperature, but of the relative concentrations of these molecules. So if you change the relative concentration and keep pressure and temperature fixed, you can change the structure. You can change the organization of these molecules. So this is only to show you that in terms of molar percent, a liquid crystal, a lyotropic liquid crystal is water. 94% in terms of mole. And the rest about 5% here or 6%, is soap, sodium disulfate, and decanol, which is the alcohol. Well, in the structures that we can have, the uniaxial nematics, as we saw this morning. So these basic units, you can imagine, or the micelles in the case of lyotropics, or a molecule. For example, the stick-like molecules that we mentioned before. So this is a symmetry axis, which is called director, as we learned this morning. It's not really a vector, but it is a direction, because if you move this 180 degrees, you have essentially the same structure. This is the uniaxial nematic. You can have another very interesting structure, which was the first one discovered more than 120 years ago by a botanist, I think it's botanist, no? Heinitzer, which was investigating the cholesterol from carrots. Investigating the cholesterol from carrots, he was able to see some properties which are typical properties from solid crystals and properties also from isotropic fluids. And this is what he saw at that time, is a cholesteric arrangement of these basic units. Imagine that you have a nematic plane, and from one to another, you turn the director, okay? So you have a pitch of an helix, of a helical structure around this direction. This is one very interesting application of liquid crystals, because this pitch is in the order of magnitude of the visible light. So it's possible to modify the pitch as a function of the temperature, and you have white light coming here and light of a particular color being reflected. This is used, if you saw probably, in some thermometers that you put in the head of children. Have you seen that before? You put a stick in the children, and depending on the temperature, you see the number. So this is a cholesteric liquid crystal. The temperature of the head of the children will arrange this pitch here, and will reflect from the white light, the light of that color, and you see the temperature. So this is a technological application of this structure. We have other structures. For example, the ismatic structure, ismatic A, ismatic C. Ismatic A, you have this one-dimensional crystal which is coexistent with a big dimensional fluid, big dimensional liquid. Okay, there is no ordering. If you take one plane like this, all the centers of mass has no positional ordering. But if you go from one layer to the other, you have a typical order lattice parameter. So it's a one-dimensional solid, crystalline solid, coexistence with a two-dimensional isotropic liquid. This is ismatic structure. If you make a small tilt in this basic orientation of this basic units for an angle theta, you have another type of ismatic phase, ismatic B phase, and you can have different types also. And we can change from one to other, changing the temperature, or in the case of lyotropic, temperature and or the relative concentrations of the different compounds. And this is something that I would like to show you. This is a typical phase diagram. Let's think like that. This is a soap, potassium laurate, an alcohol, and water. Only this. Let us fix the concentration of this alcohol here. So we can change the concentration of the soap and change the temperature. This phase diagram is extremely rich. You have two types of uniaxial nematic phase. One of them with the symmetry of a prolate ellipsoid. Another one here with the symmetry of an oblate ellipsoid. Okay? And in the middle, you have a biaxial phase. These lines here are second order transition lines. Okay, you have no jump in the order parameter. That goes directly to 0 with a tangent which depends on the model you consider here. But second order lines. And we have isotropic phase here, and this one I will left for some minutes after. So this point here is a very interesting point. It's a junction of two second order lines and a first order line. First order transition line from nematic to isotropic. It's called a Landau point, which is interesting from the point of view of statistical mechanics, is that you can measure the order parameter extremely easy in this type of systems, because what we are measuring is the birefringence. Birefringence is the order parameter. So we can investigate this point. Is one of the unique Landau points accessible from the experimental point of view. In lyotropics, we have this. Well, as I told you, this phase here at lower temperature is also an isotropic phase. Okay? Well, an isotropic phase at high temperature, no problem. We understand that. You have some basic units which are oriented. You give energy to them. They lose this orientation. It's isotropic phase at higher temperature. But look that. We have an isotropic phase at lower temperature. So there is something very special in this phase diagram. Okay? The problem is that it's possible to change the shape anisotropy of these micelles as a function of the temperature. For example, imagine that you have this ellipsoid in the ND phase. Okay? If this ellipsoid, if you lower down the temperature, becomes more spherical, at that temperature it can be completely disoriented and give you an isotropic phase. This is not possible in thermotropics because the molecules cannot change their shape anisotropy this high as a function of the temperature. But the micelles are smooth objects, so they can change this shape anisotropy as a function of the temperature, and we are able to have also this isotropic phase at lower temperature, which seems to be a contradiction. Well, these nematic symmetries, as I told you before, in the case of the calamitic phase, you have an ellipsoid here. This principal axis is the uniaxial axis here. So if you have these three axes in the laboratory frame of the laboratory, N2 is equal to N3, okay? N2 and N3 are equal, and N1 is different from the others. So we have one birefringence only. If you have the discotic phase, if you take this, you squeeze, you have now N1 equal to N2, different from N3. It's a birefringent also, but just one birefringence. And in the case of the biaxial phase, what you have is essentially, if you take this ellipsoid, you flatten this in one direction. So you have essentially a triaxial ellipsoid, okay, with three different semi-axes. And this is what we have in the case of the lyotropics with the three different index of refraction present. So we have two birefringens that can be measured. It's the only example we have in the nature with liquids with these properties. Well, so it's to awaken. If we take into account a lyotropic mixture, for example, a very simple lyotropic mixture, only water and one type of amphiphilic molecule, only this. And we can investigate the thermodiffusion, the Soré coefficient, for example, of all the components of this mixture. One thing that I will show you in a while is that it's possible, using the Soré coefficient, to measure something which is very important, which is the critical micellar concentration. We have different methods to do that. But I will show you that it's possible to measure this critical micellar concentration, investigating the thermodiffusion of the different compounds of the lyotropic mixture. Well, in terms of equations, I will pass fast by this because after feijoada, I believe that nobody is so ready to do this. But in this paper I showed you before, we are able to find all of these equations. So this is, in the case you have, for example, two solutes and one solvent. This is the current of one of these solutes. This is the current of the other solute. It depends on the ordinary diffusion coefficient that we know. The Sorel coefficient is embedded in this term here, so we can construct this current of particles, and in the steady state we will impose that this current are zero. Okay? So doing that, we are able to define this Sorel contribution, what we call the Sorel function of the solute number 1 and solute number 2. And what is interesting, if you look at that expression here, is the Sorel effect or the Sorel coefficient of the solute number 1 is also affected by the presence of number 2, which is there. This, if you imagine, for example, that you have alcohol, water, and, for example, a colloidal particle. The movement of the alcohol, if you impose a thermal gradient, will be influenced by the presence of the colloid, and vice versa. So we have to take into account the diffusion coefficients, the coupling, which couples the solute number 1 with solute number 2, and essentially what we will be able to measure is this function here, and after that we will have to make some assumptions to get the contribution of each of the solutes in our sample. So the Sorel coefficient, or the Sorel, which was proposed by Charles Sorel at that time, is essentially the ratio between this dt, which was present in the first equation, and the usual diffusion coefficient here. It has the dimension of temperature Kelvin minus 1. Okay? So this is what I told you before. If one component is affected by the presence of the others, and we have to take into account all of this stuff to have a correct description of the phenomenon. So remember the complicated letter that I showed you in one of the first slides, which take into account essentially all the Sorel effects here, the modification of the index of refraction with respect to the concentration of this particular solute. So it's possible to show that within certain approximations, we have a straight line for this parameter, where A here is the linear contribution here is essentially dependent on the Sorel coefficient of this parameter, and B, you have the contribution, crossed contributions here. And we don't know the Sorel effect here, the Sorel coefficient here also we don't know. And these terms, from our experiment, our Z-scan experiment, we don't know. But if we are able to measure this parameter and fit a straight line here, from this, the linear contribution and the angular contribution, we are able to have access to these different unknowns that we have. In the case of lyotropics, we have a small difficulty in the case of this experiment. The absorption, the linear absorption of water and the soap in this wavelength, which is 532 nanometers, wavelength is very low. So to improve the absorption of the sample, we can put some dyes. And these dyes will absorb the energy, and this energy will be delivered to the entire medium. So this is a way we can use to improve the absorption of the sample. You put a dye, this dye will absorb the light, this light will be transferred to the medium completely in the form of heat. But the important point is that the concentration of this dye should be extremely small, not to be introduced in your calculation, because all the other concentration of soap and micelles will be much higher than this doping that we do with the Congo red. One possibility to overcome this difficulty is to change a different wavelength. So if you change the wavelength of the laser, it's not necessary here. But in this case, only to give you an idea of what we can do if we don't have a laser with a broad spectrum, we can put a dye and you can have essentially the same results. So if you look what we have before and after the critical micellar concentration as a function of the concentration of soap, in this side you have water, you have Congo red, and you have free potassium laurate molecules. Okay? If you go to the other side, if you cross the critical micellar concentrations, you have water, you have the dye, you have the free molecules, but also you have micelles present. So in this solvent here, you have at least 3 solutes in this side and 2 solutes in that side. If you take into account that this concentration is much smaller than that one, you can put it out and you can measure the Soret properties of free molecules. After that, you have the concentration of free molecules here, essentially the same. All the properties measured here can be applied there. The Congo red is a small amount that will be neglected at that time, and you have the micelle. So essentially you have 2 solutes and 1 solvent. In pictures, you can see that if you increase the free potassium laurate molecules, which is sketched here, you have only molecules, and after that, when you cross this line, you start to have a concentration of molecules and micelles which coexist. Okay? And we will impose this thermal gradient and we will see what happens. So I showed you this before. It's not necessary to repeat it here. Okay, we will make the same experiment, changing the position of the sample, measuring as a function of the time the transmittance, and after that we have some kind of z-scan curve, and with this z-scan curve we can find the contributions of all the different solutes in this solvent. Well, this is what we measured independently in a solution, essentially Congo red in water. Only Congo red in water, so 1 solute and 1 solvent. It's possible to put these numbers in terms of the Soret coefficient, as I told you, in Kelvin -1, very small value. The contribution of Cn, remember, Cn takes into account the population length. Okay? We measured this in the femtosecond time regime, and we saw that the contribution of the population length in the Soret signal is very small, and we can neglect this also. Now is the measurement. So these points here are essentially the measurement of the normalized transmittance as a function of Z here. Here is for concentration of potassium laurate smaller than the critical micellar concentration. This contribution is the contribution of the population length, thermal length, and Soret length. And if you want to separate both of them, this is the contribution of the Soret, okay? Putting this arbitrary 0 only to show you what's the contribution of the Soret effect. And that one is the contribution of the thermal length. So thermal length is much more important here than the Soret contribution. So in our model, we are able to separate all of this. We measure these points. We have this fit here, and this fit is essentially a sum of the contribution of the 3 different lengths which are formed. Nonlinear from the electrons, thermal length, and finally the Soret coefficient. Well, this is the complicated letter that I don't know exactly the name. Zeta. Theta. Is theta? Oh, good. So it's theta. Maiúsculo. Okay, capital theta. our friends, your friends. Well, these are the points which measure essentially this straight line I showed you before. So before the CMC and after the CMC, you have this type of behavior. So it is possible to determine exactly what is the position of the CMC in the mass fraction of potassium laurate, for example. It's a new method. It's not the best method. I don't say that. I say that the measurement of the Sorel coefficient in this particular medium gives to us an information about the critical micellar concentration, which is not so bad, the determination here. If you compare with the CMC determined by independent techniques, this is pretty good. It's not bad. So with this, you are able to determine these parameters in blue, measuring essentially the linear and angular coefficient of the straight lines. You have these parameters. You have the Sorel coefficient of the Congo red. You can have the Sorel coefficient here of the molecules. Okay, so the molecules you are and you have also the Sorel coefficient of micelles. I'll go faster here because it's not so important. If you are interested, you can see this in the paper. But what is interesting is that the Sorel coefficient of the potassium laurate molecule, free molecules, is negative, so it's thermophilic. And when they are packed in micelles, they change the signal here, the sign here. It's positive, it's thermophobic. So the molecules itself has a particular behavior in the case of the Sorel effect. And when you put them together in water, they change the sign of this Sorel coefficient. One thermophobic and another thermophilic. So it's possible with the Z-scan experiment have access to this type of parameters. And one measurement, this is important to say, a Z-scan measurement you take about 1 or 2 hours to make. 1 or 2 hours. If you try to measure the Sorel coefficient with the usual thermal gradient cells, you need, for example, some weeks or months to have a migration of these molecules and after measure the concentration. Here, due to the small time we use, milliseconds, we are able to measure this in about 1 hour. Please.
Can you say anything about physics behind this change of sign? Do you know why this happens?
What we believe, what is happening. When you change the temperature in a micelle, what happens is that you have the double layer around the micelle, the bilayer, for example. And the charges which are there depends mostly on the temperature. Because if you change the temperature, the equilibrium between the bulk and the surface of the micelles change. So in the case of the thermophobic, it means that the micelles tend to go away from the hot part of the sample. So what we believe what is happening is that you are charging more the micelles, so the electrostatic interaction between them put them apart. In the case of the free molecules, you don't. Yes, sure. It's not in this explanation, in this guess I am giving, it depends on the presence of other micelles. It's not only a property of a single micelle. Okay. Pardon?
So that the interpretation to be true, the interaction between micelles should have a very large range because just above CMC, micelles are pretty dilute.
Yes, it's electrostatic origins. It's long range because the micelles has a net charge. No, it's a guess. I cannot tell you that it's.
They're saying that the chemical potential is lowering when it's thermophilic, is lowering this temperature because there is a.
Could be also. Sure. Yeah. At this level, I cannot say that. The chemical potential, the change in the chemical potential. Yeah, it's another possibility. Yeah. Well, so it's possible to have this, we have access to the Sorel coefficient there. This is another method. It's very sensitive to the measurement of the, oops, sorry. It's very sensitive to measure the critical micellar concentration. Okay. Okay. And something that is very interesting that I would like to propose you as the fifth exercise to send me is that if you take into account the thermodiffusion, the thermophoresis, and the differential diffusion, this tells us something about the movement of the basic units, the molecules and the micelles. And this is a possible conclusion, that they always goes towards the warm region of the cell. So I would like you to discuss this expression for me. I will send you the paper where this is discussed in some details, but I would like you to go a little bit deep into the details to understand the difference between thermodiffusion, thermophoresis, and the differential diffusion. Okay? Well, some people involved, very good. So now is at the end, I would like to discuss with you the third family of complex fluids that we can investigate with these techniques, which is the human lipoproteins. Okay, it's essentially the cholesterol we have in our blood. Well, why we will investigate this? Okay, for the fundamental reasons, it's okay, but if you take into account what the World Health Organization says about the amount of cholesterol that we have in our blood, they say that if you have high levels of cholesterol in a blood exam, high levels of low density lipoproteins, LDL, triglyceride levels high, low levels of high density lipoprotein, you have a risk to develop a problem in your heart. Okay? Heart disease, ischemic strokes, etc., etc. Well, so this is interesting. It's interesting for us to study. So when you look at this, which is written here, you see that LDL is a devil. So you have to get rid of it. In the other hand, HDL is a good guy, the bad guy and the good guy. So you have to have a lot of HDL in your blood and very small amount of LDL in your blood. So this is what is the translation of this written in English to picture. Well, let us go a little bit deep into these details. This is the cholesterol. This is the molecule, cholesterol. And the cholesterol is essential for the metabolism of the cells. If you don't have cholesterol, you are not alive. So we need cholesterol. The cholesterol is produced essentially in the liver. We eat a little bit of cholesterol, probably in porcão. We ate a lot of cholesterol molecules, but this is not the principal source of cholesterol. The cholesterol which is produced in our liver is the real source of cholesterol. But the problem of this molecule is that it's not soluble in polar solvents like our blood. So we have a problem. We have the cholesterol produced in our liver, and it has to be conducted, for example, to the head by our blood. And how we can do that? In this molecule, if you put in water, it goes down. Okay. So nature has a problem. He created some kind of carrier of cholesterol. I call this a nanosubmarine. You will see why it's a nanosubmarine. These lipoproteins, low density lipoproteins and high density lipoproteins, are the main carrier of the cholesterol in our blood. So what is this? Why is this important? Well, this is our lifestyle, okay? At this position here, we have a lot of problems. No? People may die, heart, so this is not good, but this could be worse. If you take into account this here is an amount of fat around the basic organs, the most important organs we have. Okay? Because of that, we have diabetes in the higher age we have. So the lifestyle of us tells us that we have to make something. So this is the nanosubmarine which the nature did to carry the cholesterol from the liver to the different parts of the body. And this submarine is now soluble in water. This is the HDL, and the LDL is here. Bad guy. It's not the bad guy, you will see. So what is a particle of LDL? If you remember a micelle in a allotropic liquid crystal, you had amphiphilic molecules around and the paraffinic chains in the central part. Okay? LDL is essentially that. You have phospholipids around. Okay? You have these blue heads are the polar heads of a phospholipid. Inside that region here are two carbonic chains, so you have a head and two carbonic chains. Okay? And the cholesterol, the esters of cholesterol, the triglycerides are inside. So this part is soluble in water, in blood, and what is inside is not soluble, but it can be transported from one part of the body to the other. This is a protein. Each LDL has one protein attached to it. And this will be recognized by our cells and will be internalized, and you will see in a while what is the trip of the LDL. Please, forget this part and concentrate here. These are electron microscopy pictures of LDL. The typical dimension here is about 20 nanometers. So the diameter of an LDL particle, 20 nanometers. And here is the HDL particle, okay? Much smaller than that one, more dense. Because of this, it's called high density lipoprotein. The LDL makes the direct transport of cholesterol from the liver to the different cells, and what is the excess of cholesterol is removed from the body to the liver, again, by the HDL particle. So this is a direct transport of cholesterol, and this is a reverse transport of cholesterol. The trip of this nanosubmarine in the blood is the following. It's in the blood. These are some receptors in the surface of the cell. It recognizes the LDL particles, put the LDL inside the cell. It breaks the LDL. The cholesterol is delivered, produces more of these receptors, make everything necessary with cholesterol, the metabolism of the cell. And after that, this excess of cholesterol will be put back by the other nanosubmarine, which is the HDL. So, if your LDL is okay, is not modified by an external agent, it will be recognized, it will be internalized, the excess will be removed by HDL, there's no problem, okay? But the problem arises when this receptor could not recognize the LDL particle. For example, if a bacteria or a free radical changes the structure and the property of this particle. If it's no more recognized by the receptor, our antibodies will try to fight against this modified LDL. And this is the formation of the plaques in the heart, in the arteries. You have can give a stroke, can give infarctus of myocardium. So the production of plaques in our body is essentially a fight of our antibodies against something that it did not recognize. It's our immunological system. Well, so it's interesting to identify or to differentiate the LDL modified from the LDL native, a good LDL. So when we do a blood exam, in any lab actually around the world, what they give you is the amount of LDL, total amount. They don't give you the amount of modified LDL. And this is what is important. Because of that, sometimes you see a guy which is very thin, no fat, nothing, and dies with an infarctus. Why it happens? It has very high amount of modified LDL. So this is important, to know this number, this quantification of modified LDL. The total number is not the important number. Obviously, if you reduce the total number, you reduce proportionally the amount of LDL which is bad in your body. Because of this, we take statins, rosuvastatin, Crestor, Lipitor, etc., to low down the total amount of LDL. But this is not good. The good is to fight against the bad LDL. And at the end, I will tell you something which is very interesting, because we had some patients in Sao Paulo, very thin people, nothing of grass in the body, but with problem of very large amount of plaques in other parts of the body. And we could demonstrate that they had periodontitis. You know what is periodontitis? It's a problem in the gem. You know, it goes up, and it is an inflammation of the gums. No, it's this. And what happens is that it's caused by a bacteria, and this bacteria goes inside the blood of these people, and the fight of the immunological system against this bacteria changes the aspect of LDL. So people which has this periodontitis has a very high possibility to develop heart disease. And to cure this, only wash the teeth. That's the only treatment we have. Only wash. Okay? It's very important. This. Well, this is something that we can do in vitro, which mimics what happens in our body. This is the normal state of LDL, now much more concentrated, and we induce the modification of the LDL, mimic the action of a free radical. The free radicals we have in our body, okay? So when the free radicals attack these LDL particles, they oxidate these particles, we form these aggregates, these linear fractals, which are formal vesicles. This type of particles will not be recognized anymore by our immunological system. It will believe this is really bad, so the plaque will be formed. So we took blood from people. We call them the normolipidemic blood donors. They are volunteers, so they give blood for us. Sometimes it's very difficult to find volunteers, so we take some guys from the lab, no problem. It's very good. It's very good. And after that, with this blood, we make ultracentrifugation of this blood. This is not made in the Institute of Physics, so this part is made in the hospital. After that, we go to the Institute of Biomedicine. So you see that there's some type of work which is multidisciplinary. We cannot do this in physics only. We'll do physics after. Until now, we were in the hospital. Now we are in the chemistry department or the immunological department to separate by ultracentrifugation the different types of particles. Here's the HDL, LDL, and the rest. And we can remove the LDL from here. If they are normolipidemic, so good guys without illness, we have native LDL. And with this native LDL, we can induce or mimic modifications putting free radicals. And we investigate the response of these particles. This is something that we did with free radicals originated from copper. We can do this also with iron. We have a lot of iron in our blood. So part of them may induce this modification of LDL. So these are the native LDL. We started like this. After that, you see that the copper oxidation of LDL changed completely the shape and the size, the distribution of the particles. Iron here also. And this is only to show you that we made some investigations if we have fusion or clustering of particles. And we were able to show that we have essentially clustering and not fusion of different LDL particles. Well, this is the linear absorption. The lady there asked me how to measure the...
Yeah, I asked before with isotopic or you usually measure the transference and you use the Lambert-B equation to convert the absorption, the thermal from absorption. But it also only works with bulk and not with thin films.
Yeah. Okay. So this is also the absorbance, the linear absorbance as a function of the wavelength here. So you see the difference between the normal LDL, the native LDL, and the copper oxidized LDL. Okay. So with this, we can measure the Z-scan in the thermal regime. So this is the signal of the native LDL. This is the copper LDL. You see almost nothing, almost flat here. And the other one is the iron. I think. Sorry about that. Take this out. Okay. So you see that the thermal optical coefficient can change drastically from one to the other, or other parameters which are enclosed in this theta. Remember the theta parameter. The theta parameter had a lot of different types of contributions. One is dN over dt, the other is the density, the other is the heat capacity, and the final one is the conductivity, the thermal diffusivity or thermal conductivity of the sample. So in this case, we measure theta as a function of the good LDL concentration. If you have this type of curve and what you have in your blood exam, you remove one from the other, you have how much or how many bad LDL you have in your blood. So this is a method to quantify the good LDL you have in your blood, because the bad has a flat signal. We have no signal for the bad LDL. So if you take this number and the number which are in your blood test, you have essentially the number of bad LDL you have in your blood. I don't know if I have theta here. It's theta, but it's not written. Well, so we, with the people from the chemistry department, we investigate what is happening in the process of oxidation of this LDL, and we were able to understand what is going on, what is the effect of these free radicals in the structure of these particles, in the shape of these particles, modification of the shape, and we cross this information with a small angle X-ray scattering, which give us a very good information about the shape and the shape anisotropy of the particle. So this is what we did, and we are able to measure this parameter here. Let me see if I have another. Okay. So this is, sorry. Okay. This is the thermal diffusivity of the sample, which is a function of the oxidation time. So what we see here is that the normal or native LDL is almost, does not conduct heat too much, but if you oxidize this particle, this thermal diffusivity now increases a lot. So probably this could be in the future one of the factors of risk that we can measure in our blood to see the amount of LDL modified particles. Okay, so it's some type of contribution from the physics to the applied medicine. If you measure this parameter in your blood, you can have an idea of how many bad LDLs you have there. We have to explain to the physicians, to the what a thermodiffuse is, but no problem, I can try to do that. Well, just to finish, I have 3 or 4 minutes. I put this heart here. The heart is because LDL and nothing related to heart. So some conclusions that we can have from here. That this oxidation with copper and iron changes the morphology of these particles, changes the size, shape distribution, may form aggregates, and these new objects will not be recognized by ourselves. So plaques should be formed. This copper mediated, as you saw, is the strongest way to modify your LDL particles. The iron is not so efficient. The iron is a little less dangerous than the copper. So copper, if you have free radicals, copper in the blood, it's very bad because this promotes the highest modifications of the shape anisotropy, the shape, and the aggregation. No fusion of particles were detected. For example, this wasn't one doubt. Probably the particles fused and they became very large, and this cannot be identified by the receptor because it could not fit. This is not the problem. The problem is that they aggregate and they can attach to a small part of this aggregate, so the plate will be formed in these conditions. As I told you, the copper oxidation is extremely drastic, so it's something that we have to avoid to have this type of free radicals in our blood. So these are people involved, people from different types of backgrounds, physicians, physicists, mathematicians, to define, for example, the number of patients we need to have a good conclusion. For us physicists, we do an experiment, okay, we extrapolate this for everything, but for medicine, it's completely different. They say we have to take, for example, 110 patients, we have to have controls, so it's we need some type of support from the mathematics to do that, statistics related to medicine. Okay, I think it's okay. So thank you very much for questions, comments.
Well, linear properties are not good enough.
No. Okay, I can send you after 2 or 3 papers, 3 references, telling something about the Sorel coefficient and the different time scales and this part of LDL. So if you're interested, we can keep in contact after. Okay? Probably they have to go to the